So good morning. Welcome. Most of you know me, but for those of you who don't know me, I am Fabian Michelangeli, the Abbess Curator for Tropical Botany at the Institute of Systematic Botany at the New Botanical Garden. And it's my pleasure today to stand in for Barbara Ambrose, who should have been doing this introduction for our speaker today. Sebastiano Nigris. Sebastiano has been here for 6 months and he lives in 3 days back to Italy. And this is a little bit of a bittersweet introduction because he's been a great citizen, fun person to have in the lab, has been very collaborative, has become a complete part of our research family in the lab in this last 6 months, in spite of all the issues we have had with COVID and restrictions and all of that. I want to remind you that this webinar is being recorded and that you can access the chat function to, the question and answer function for your questions. Please don't use the chat, so that's a little bit different. And live captions are enabled by clicking the closed caption button, then show subtitles, and then view the full transcript. So Sebastiano will be talking today about the importance. This is the importance of studying Gymnosperms to unravel seed plants evolution. Sebastiano obtained first his master and then his PhD at the University of Padua in Italy. And then he went on to do a postdoc also in the University of Padua. In between, he spent also some time in Wageningen in the Netherlands doing some research. His first research was on the plant, the bacteria associated with the roots and stems and leaves of grapes. And then he started working more on development and reproductive structures in Nymphea, in water lilies, of which he spoke about yesterday or 2 days ago. And now he is more working on the topic that he will be studying. He also has experience working with ancient DNAs from all herbarium specimens in Padua, but in this case with fungi. So he has really spanned a lot of different things. One of the things I find fascinating is that he works at a botanical garden that is over 800 years old. We've pride ourselves here in New York of having one of the oldest botanical gardens in the US. Well, this is almost an order of magnitude older, and is really truly an incredible institution. So again, I don't want to finish this introduction because I don't want Sebastiano to leave, but welcome and please go ahead.
So thank you, Fabian, for the warm and nice introduction. So, okay, can you see my presentation?
Not yet.
Okay, not yet. What's happening?
There it is.
Okay. So thank you. Yeah, thank you, Fabian, for the nice introduction, from the kind introduction. Thank you all to give me the possibility to talk about why is so important and why so sexy the study of Gymnosperm to understand somehow plant evolution. Wait. Okay, so one of the fundamental questions when considering the study of plant evolution is devoted to understand which are those molecular mechanisms that can regulate the patterns in ovule and seed development. And which are the molecular mechanisms that led to the evolution of these reproductive structures. And these reproductive structures are fundamental for the plant development and for the plant evolution. Because if we consider that reproduction mechanism, especially those mechanisms that involve ovules, flowers, fruits, and seeds, have a great impact on the ecology of the species. And so they are kind of part of the driving force that is actually driving the evolution to the species, to the plant species that we know. So if we give a look to the whole evolution of plants, we can easily understand, and if we consider what we know from plants, it's easy to understand that most of the information that we know about ovule and seed development comes from angiosperms, and particularly from a bunch of few model species like Arabidopsis, tomato, rice. From these plants, we have a lot of information about the tissue development, about the genes that are involved in the development of these tissues. Yeah, all this information are coming thanks to the study of mutants. And there are some nice reverse genetic tools that could help the researchers to explain which genes are involved, how the genes can interact in the formation of the gametophyte, in the formation of the two integuments that protect the ovule during the development, and eventually they can explain which genes are involved in the development of the seed coat. So we know a lot of mechanisms subtending the formation of this reproductive structure in angiosperm. And we also know with a quite good detail, even if we are knowing even more, but we have a good knowledge about the process of pollination and fertilization of the ovule. And we know that in angiosperms are often very close in time. Pollination and the fertilization is just after pollination. After fertilization, we have the transformation of the ovule in seed. So these processes during the time have got an important light because basically most of the human diet, right, is based on seeds. And this is so, yeah, it's so clear that that we know about this mechanism of ovule development and seed development. Most we know about this mechanism, better we can act to, how can I say, to improve somehow food production, food safety, with the classic techniques of plant breeding to obtain new varieties with better characteristics, or with the modern technique of genome editing. So when I talk to my students, I always give an example. So when most of the grape that you find at the supermarket now, it's seedless. So these berries, these seedless berries, are derived from plants that have a single mutation in a regulative gene that is seed stick. It's a matchbox gene. And so you can see that the study of developmental processes for seed production and for ovule development and seed production are of great importance also for the crops and the fruit and whatever we use for human nutrition. But if we go back to the plant evolution, there are some information also on reproductive patterns of those plants that we can call lower plants. So there are some emerging model plants, like some moss species, lycophytes, and ferns. And the researchers are developing reverse genetics techniques and functional genetic techniques to understand in these plants the mechanism of the reproduction, the mechanism of the gametophyte and the sporophyte differentiation, which genes are involved and in these processes. So we are starting to have a good base of knowledge even in these lower, let's say, lower plants. And what unfortunately is still missing so far, it's a good knowledge about the developmental mechanism, the developmental processes in most of the gymnosperms. This is because gymnosperms are, let's say, difficult plants. Most of them are trees, and probably because that's my personal opinion, probably because gymnosperms are not directly related to food questions. So they are still related to a small botanic interest. However, if you just see the evolution of plants, the knowledge of this plant, the knowledge of the molecular mechanism that led to the evolution, to the development of the ovule and the seed, beside to be of great interest by themselves, can somehow help to understand the evolutionary pathways that led to the formation to the seed, to the appearance to the seed in the plant species. Because, gymnosperms are spermatophytes, and because the ancestral gymnosperm species developed for the first time the seed as a mean of embryo dispersal in the environment. So gymnosperms have a great biodiversity. Most of them are trees, of course, are seed plants. There are four groups of gymnosperm that are cicads, Ginkgo. I would say Ginkgo. I know that you say Ginkgo, but I will say Ginkgo for all the talk. Conifers and Nietales. The evolutionary history and the phylogenetic relations are not still completely clear. There is a good fossil record, and with the availability of many genomes now, researchers through phylogenetic analysis are able to reconstruct the phylogenetic relationship among this group. But still, there are some interpretations, there are some hypotheses, and the landscape about phylogenetic relations is not completely clear. And even if the name from the ancient Greek means naked seeds, gymnosperm, however, protect the seeds with dry or fleshy structures. So, in fact, gymnosperms, of course, can't have botanical fruit as we consider the fruit, because the fruit is transformation is coming from a transformation of the flower after fertilization. But they can still have fleshy structures associated to the seed in order to protect the seed or to improve the seed dispersal by frugivory. So these structures can have different anatomical origins, can be a fleshy integument, can be a fleshy receptacle, can be an aril that enclose the seed, like in the case of Taxus, baccata, or can be some fleshy bracts that protect the seeds, like in case of Ephedra species. So with the research group which I belong, we started to focus our attention in gymnosperms, ovule and seed development. So it's a relative young research group. We started in 2017. The principal investigator is Professor Baldan of the Department of Biology of the University of Padova. And at the beginning, we start investigating early diverging angiosperm flower development, development, and then we move to study the ovule and seed development in Gymnosperms with an evolutionary point of view. So the first plant that we start to working with is Ginkgo Biloba, and that's the reason why we are in contact with Barbara Ambrose and Dennis Stevenson, because we realized that we were working with on similar aspects of the development and the biology of this plant. So Ginkgo biloba is the only extant species of Ginkgo phyta. There are many fossils describing different species, but still, from the fossil analysis, we can say that essentially this plant has been unchanged for more than 2 million years. So the genome, it's a huge genome. It's 10.61 gigabytes, and from the genome draft that is available in the public database, we know there are at least 41,000 genes. And from the analysis of this genome, we know that there are at least 2 whole genome duplications. So the genome is huge, is 80 times the genome of Arabidopsis. So it's easy to understand how such a complex genetic background can even discourage from the beginning researchers or from founding agencies to start a research on this plant. But well, I think it worth to focus on, because there are some mechanisms that can really help to understand how the evolution worked during plant evolution. So the male plants produce small pollen cones that we call catkins, and the female plants produce ovules at the end of a stalk. So usually are 2 ovules, but we can find also 3 ovules, even 4 ovules at the end of the stalk, inserted in a collar. So ovule and seed development in Ginkgo biloba is essentially a long story if we compare the process with the process of angiosperms. So let's start the process in winter. So in winter, buds are at the end of the characteristic short branches that we call brachyblasts, are well enclosed with scales. As you can see here, this is the terminal bud of brachyblasts. So inside these coriaceous scales, we can already find in winter the primordia of the vegetative organs, so the primordia of the leaves, and the primordia of the reproductive organs. So in this case, ovules. And from the picture, you can already recognize the characteristic shape with the 2 ovule primordia at the end of the stalk. So at early spring, buds open, so the ovules are exposed in the atmosphere, and they start grow. They start the visible development process. So at the beginning of spring, the ovules are almost ready for the pollination. In fact, a few weeks after the buds opening, we can observe the pollination drop. This drop is extruded by the ovule in order to catch the pollen from the atmosphere. So after pollination, that occurs, in Padova, it occurs usually at the beginning of April, according with the weather conditions. So after this pollination, the ovule goes through dramatic morphological changes all along the summer. at the end of the summer, we have the fertilization, when the when the male gametophyte inside the ovule can release 2 sperm cells that can fertilize 1 egg cell. So in autumn, the seeds, so fertilized ovule are now seeds, assume the characteristic morphology that we observe when the seeds from Ginkgo fall apart. They are, how can I say, with a bad smells, with a soft integument, and inside a lignified coriaceous tissue that enclose the embryo. So when seeds are ready for the dispersal, at the end of the brachyblasts, there are already the buds for the following year. So it's a kind of cycle that starts from the winter and finish to the winter. So what we have done in my lab is to build up an atlas to orient people and to help to people to understand the developmental process. And what we thought and what we tried to do is to set some stages that were not related to the calendar, to the season, like April, May, but stages that are recognizable, analyzing some landmark transformation inside or outside the ovule. So I will show you the main stages, the main recognizable stages of this process. So in stage 2, you can see that ovule primordia that are still enclosing winter buds, so everything was still inside the buds, present themselves like with the characteristic shape, like the end of a bone. So there are no signs of integument development, but we just see the 2 primordia standing on the stalk. If we go to the stage 3, now it's more clear the shape of the ovule, and it's clear that there is a differentiation between the nucellus, this rounded structure here, and the integument that is starting the differentiation. It's also possible to observe more clearly that there is a kind of sulcus between the 2 distal portion of the structure. So if we move to the stage 4, we can see that the integument start to grow to enclose the nucellus to get to the final shape of the ovule. And this is more recognizable if we observe what we call stage 5, when the integument has completely enclosed the nucellus, overgrowing on it. And at the stage 5, we can observe that there is the first signs, there are the first signs of color differentiation. So beside the external, let's say, macroscopic modification of the ovule, some stages are defined by what's happening inside the ovule. So, for example, stage 6, at stage 6, we can observe the meiosis of the MMC and the mitosis of the functional megaspore that will form the female gametophyte during the development of the ovule. So now the ovule is ready for the pollination. In fact, at stage 8, we can observe the pollination. So the pollination drop, it's a common mechanism in Gymnosperm to catch the pollen from the outside. So the pollen drop is extruded several times during a few days, and is extruded and withdrawn inside the ovule, until the ovule somehow feel the right pollen. So the pollination drop is extruded from the micropyle that you can see here with ACM image. So it's basically a channel that connects the external environment to the pollen chamber inside the ovule. So stage 8 is pollination. Stage 9 is when the female gametophytes start to grow. At the beginning, as a free nuclei structure, so is not still cellularized, but there are some free nuclei inside the space where the female gametophyte will be formed. And we start to observe the integument differentiation. So differently from angiosperms that have two integuments, Ginosperms, and so Ginkgo, has just one integument. But we can already observe that there is a morphological and a correlated functional modification, differentiation in 3 layers. These 3 layers will be the three characteristic layers that will be observed in the Ginkgo seed. So there is an endotesta that will be degraded in like a papery layer. There is a middle sclerotesta that will be lignified at the end of the development. And there is the outer layer that is the fleshy sarcotesta. So at stage 10, we can observe the cellularization of the female gametophyte, and we can also observe macroscopically that there is a differentiation in the integument, in the fleshy region, the outer one, and the part that will be lignified like an almond here, that enclose the rest of the nucellas and the female gametophyte. This differentiation is much more clear if we stain with a staining that stain lignified tissues. So if we go farther with the stage, we can see at the stage 12 the archegonia formation. Archegonia are formed here in the apical part of the female gametophyte, and will differentiate in the egg cell. And this stage is in midsummer, as I told you before. So the last stage that we set is the fecundation. So one swimming sperm cell released from the male gametophyte can reach one of the 2 archegonia, can fecundate, can fertilize the egg cell, and then we can observe the embryo development. This is an early embryo development observed in late summer. So to understand which are the processes and which is the trigger that that trigger the ovule development, we decide to perform 3 RNA secs, 3 RNA sequencings to see all the genes involved in this process of development around the pollination drop. Because our hypothesis is the pollen arrival can trigger the further development of the ovule. So we analyze a pre-pollination stage, a pollination drop stage, and 3 pollination post-pollination stages. So what we observed from the data analysis, from the analysis of the genes expressed in these 3 conditions, is that many genes that we thought that were involved in the process because involved and activated in Arabidopsis after fertilization, so genes responsible to the transition from ovule development to seed development, were actually not activated in Ginkgo upon pollination. So what we have observed, thanks to the RNA-seq and thanks also to a metabolomic analysis, is that after pollination in Ginkgo are activated some pathways relative to the transformation of the integument into the seed coat. So mainly lignin biosynthesis pathways and fatty acids biosynthesis pathways. These pathways are early activated after the pollen arrival, so just in the few days after the pollen, and these processes will lead to the formation of the three different layers of the integument. So even if there is a single integument, but after pollination there is a transformation of the integument of the layers with different morphological and functional characteristics. So what we can conclude with this experiment of RNA-seq and metabolomics is that pollination is the trigger of further ovule development and the trigger for the integument differentiation. So we plan to do, of course, further experiments, and these experiments will be focused to understand if there is a set of genes that can lead to the formation of ovules and not leaves. What we have in mind is to compare ovule primordial transcriptome and leaf primordial transcriptome to see if there is a set of genes devoted to the formation of leaves and ovules separately. So thanks to this work in Ginosperms, we could be granted with a project with a funding from the European Union that is called Ovule, and this project aims to make an investigation, comparative investigation of the conserved mechanism in the evolution of seeds and fruits and fruit-like structure of Gymnosperms. So it's a comparative research, and this project is in the context of the development of new strategies to improve food production in terms of food availability and food sustainability. So there are many partners, many in Europe. So two laboratories, two research groups in Italy, my group and the group of Lucia Colombo in Milano, the group of Barbara Ambrose here at the New York Botanical Garden, a research group in Mexico, a research group in Colombia, one in Australia and one in Brazil. And all the research groups are focused on different aspects of ovule, seed and fruit evolution and developmental mechanisms. So since also Barbara Ambrose is interested in Ginosperm evolution, Ginosperm seeds, thanks to this project, I got the possibility to work here at the New York Botanical Garden, and we decided to work on Taxus baccata ovule development. So Taxus baccata, it's of course another Ginosperm. For this plant, a recent genome, even if it is still a draft, is available. There are many transcriptome present in the public database, and we have also some transcriptome made by my research group and people here from the New York Botanical Garden. So the main characteristic that make the plant famous is the red aryl that completely enclose the seed when the seed is mature. And the red aryl is the only part not poisoning of the plant. And this is a characteristic in order to enhance seed dispersal by birds. So they can feed on the aryl and they then drop the seed around intact and they can enhance the plant, the species diffusion in the environment. So actually, just few information in terms of molecular mechanisms are available on the aerial development. But actually, so far about molecular, so genetic regulation of the ovule, nothing is known. So starting from the transcriptome analysis, I was able here to isolate some genes that we know are involved in developmental processes of the integument, of the ovule, and so on the leaves, on the seed production. So we decided to analyze two time points, one sampled at the middle of September, and one more recent at late January. So I isolate genes from the transcriptome. I characterize them by similarity. I do some phylogenetic analysis, and I can conclude that I can select these 6 genes for expression analysis. So 2 of them belong to the family of class 3 HD-ZIP, and we know that they are important in many developmental processes in plants. This family is involved in many developmental processes in plant, like Procambium differentiation, and in particular in the field, in the context of ovule development, they are involved in the adaxial abaxial, so in out symmetry and polarity of the integument. The same processes, in the same process, so polarity of the integument of angiosperm are involved these Canadi genes, that is another family of transcription factor, of growth regulators. And I was able to isolate and fully characterize 2 of these genes. While the last category of genes that I found 2 of bell genes. They are tail genes, so transcription factors involved in the nucellas and the integument growth. So I did here expression analysis through in vitro hybridization to see in ovule sections where these genes were expressed, and if there are some changes in the expression of the genes during the time. So to show you how the 2 stages that I decided to use look like, I use one apical picture of the stage 1, where we can see the nucellus that is surrounded by many different bracts, and with 2 black arrows. I pointed out these 2 bumps that are probably the initiation of the integument growth. So there is not an integument formed in this stage, but we can just see the nucellus tissues. But there are some, these probably are the integument initiation that start as 2 bumps and then will be like a ring that overgrow over the nucellus, covering at the end the whole structure. And this process is more clear here in the second stage, when we can see all the bracts enclosing the ovule, and the integument that overgrow on the nucellas almost to form the micropyle channel here at the top. So for both these stages, the average diameter of the ovule is 0.4, 0.5 millimeters. So I want to show you some pictures about in vitro hybridization experiments. So with this technique, you can literally see gene expression on your tissue, on your sections, like a color, thanks to a colorimetric reaction. And it's clear here that the two genes belonging to the same family have differences in expression. So the first one is more expressed on the top of the nucellas, and probably if we consider this the initiation of the integument grow even here. And this is less expressed in the first stage, and the expression is more located on the top of the nucella surface. So there is a different spatial regulation of the gene. If we consider the second stage, the situation changes dramatically, because the first gene, class 3 HD-ZIP1, is broadly expressed in all the structure, so even below the ovule, where the ovule is inserted in, let's say, in the female cone. And this is probably the integument overgrowing the nucellas, going to close the structure, forming the micropyle, is expressed all and strongly again on the apical surface of the nucellas. And this here, for the second gene, the class 3 HD-ZIP2, the situation changes dramatically, because there is almost no expression for these genes in the second stage. There is just only a faint signal from the nucellas. So if we observe the expression patterns of the two Cannabis genes, the first one shows a very strong signal again on the surface, on the nucellar, on the ovule structure, the nucellar structures, probably where the cells are proliferating more, where there is more cell proliferation. And the other gene is very low expressed, but still spatially located in the surface of the structure. So again, if we observe the second stage, the situation is different for both the genes, because there is a broad expression all along the nucellus that now is enclosed by the integument, and in the integument, and also for the second gene that have a stronger expression compared with the first one. So there is a kind of change of the situation. The first gene is more expressed in the first stage, and the second gene is more expressed in the second stage. So from these experiments, we can see that there is a collaboration between the genes. There is a spatial differentiation. And this is also clear for the two bell genes that I could analyze here. And it's easy to observe that in the case of the first gene, there is a low expression level at the very top of the nucella, and the expression is still low, but is more located in the second stage in the growing integument that here has already complete the growth to enclose the nucellus. And there is a stronger signal in the integument rather than in the nucellas. And in the second gene, we can see that there is an expression on the top of the nucella again, because almost all these genes are much more expressed on the top of the structure, because, yeah, my interpretation is there is the maximum amount of cell proliferation because the nucella is growing. So you have cell division, cell processes. So all these genes that are working for the development, for the growth of the structure, have the maximum of expression there at the first stage. But the situation dramatically changed at the second stage, where we have a huge expression in the second, in all the tissues of the ovule. So all these pictures can give us an idea of the spatial and temporal regulation of the functioning of these genes. And what is clear, if we can conclude integrating what we have seen in Ginkgo, what we observed in Taxus, is that in Gymnosperm, of course, there are some conserved mechanisms conserved with angiosperm, because orthologs of the gene that we analyze in Ginkgo and in Taxus are, of course, present in angiosperms, and they act during the development of the ovule and of the seed. But these genes have different timing and spatial regulation. And what also is clear from this observation of our results is that there is not a clear symmetry in ovule development, so like abaxial, adaxial, like in angiosperms, where the tegument have an inner side with some genes expressed there, and some other genes expressed in the outer part of the integument. So probably in Gymnosperm, what is most probably to be is a symmetry apical basal, where there are some processes that occur in the apical portion of the nucella of the ovule of the integument, and other processes that occurs in the basal portion of the ovule. And this is a clear and difference between angiosperm and Gymnosperm, and probably we have to take in account when we have to describe this genetic and molecular mechanism that lead to the ovule and seed formation. So what we also observed and what we understood is that there is a temporal distance between the event of pollination and the event of fertilization. And this temporal distance is actually a pro, because it allows to have like a slow motion picture of the developmental processes that from ovule to seed. So why, which is the importance to study Gymnosperm? So the importance is clear, because they are an important group of plants along all the evolution of plants, because they firstly adopted the seed for the embryo dispersion in the environment, and then because even if they are not related to any how can I say food related research, but Gymnosperm can tell us a lot about evolution of seed, and of seed. And so and also because Gymnosperm are such difficult model plants, are such difficult plants. There are no molecular tools, no genetic reverse genetic tools. So they when you work with them, you can have really high endorphin spikes. So thank you, Fabian, for the suggestion. So let me conclude with some slides about the Botanical Garden of Padova. Fabian, actually the Botanical Garden of Padova is not 800 years old. It's the university that is 800 years old. The Botanical Garden has been founded in 1545 because some professors of the university realized that for students in medicine, in pharmacy, could be much more easy to learn how to recognize the plants from living plants and not from book illustration. So the first idea of a botanical garden was a living collection of medicinal and poisoning plants for medicine students. So after that, with the time, the garden was enlarged and became one of the main center of one of the main center in Europe for the study of botany. So unfortunately, along the years, the research at the botanical garden has been slowed down, but now in the last year, we are doing our best to keep high again the research at the botanical garden. So the botanical garden was at the beginning only enclosed by the circular walls that are still now there, but at the with the time has been enlarged around the walls. So we have some historical trees. So the oldest plant now is a palm. It's a Camerops humilis. This plant was planted in 1585, so it's quite old. Probably probably for sure this plant has been seen by Galileo, and this plant has been described by Goethe, the German writer, in his books. So it always been growing inside the greenhouse. So the plant reaches the height of 11 meters. So the other two historical trees that we have is a Platanus orientalis from 1680, and the Ginkgo, that is the object of our study, that was planted in 1795. It's a male tree and with a grafted female branch, big female branch. So recently, the botanical garden has been enlarged with a new greenhouse and with a new building where our laboratories are located. And in the greenhouse, you can find all the environment present, almost all the environment present in the world and important for plants. So you can go from tropical environment to temperate environment through Mediterranean, and then you can reach the arid environment at the end of the greenhouse. So thank you a lot for your attention, and I would like to say thank you to all of you for the attention here during this talk. And in particular to Barbara Ambrose, to Tanisha Small, that helped me to perform this research, to all my collaborators in Italy, and the research group of Professor Barbara Baldan in Padova, and the research group of Professor Colombo in Milano. Thank you.
That was great. I don't know if Danielle, please, yes, here I am back. So thank you, Sebastiano. That was very, very interesting and beautiful work. And I don't know how many people realize how hard getting these in situs to work, especially not model organisms, that is. And we already have a few questions coming in the question answers, and I will read a couple to you. The first one from Damon, which is actually something I was thinking about, is what do you think is happening in the long gap between pollination and fertilization? Is there selection of pollen for the prevention of hybridization or inbreeding? And that's Damon's question. And I'm gonna add to that very little part there is, are all these gene activation cascades that happen, do they happen more when the pollen, when on pollination, or in fertilization, or is it happening throughout?
Okay, so about the first question is the okay, all along this time between pollination and fertilization, you have the grow, basically the grow and the, how can I say, differentiation, maturation of the male gametophyte. So when the pollen arrive inside the ovule, is caught from the atmosphere, is brought inside the ovule, and there is and there inside the ovule, inside the pollen chamber, the male gametophyte start to grow like a branched male gametophyte. It forms like an haustorium that grow inside the nucellas and take nutrients from the nucella tissues. And in all this period, you have the differentiation of the sperm cells, the maturation of the sperm cells, and eventually they can be released, going to the and they go to fertilize one ovule, one egg cell. Well, probably through the mechanism of the pollination drop, you can catch many types of pollen, but there are some signals that tell to the plant that the right pollen is arrived, because what we observed is that if ovules are not pollinated, the ovules are degraded. There are some phenomena of cell programmed death inside the ovule, and they eventually fall apart from the tree. So there are some mechanisms of pollen recognition and some mechanism of, let's say, species recognition. Well, and also along all the time between pollination and fertilization, there is the differentiation of the ovule. So it's not a static process, but there are so many processes that occur in that time, both morphological and molecular, that eventually lead to the fertilization.
Great. And I have another question here from Barbara. She wants to know that, as you mentioned, there is no polar expression of C3 HDC or CAN in the integument, but do you see any expression of these in the bracts? And do you have any data for the aryl part in Taxus?
Well, the expression, the bracts, is not very clear. What I observed is no expression in the bracts for the first gene, at least on the picture that I was able to analyze. And if we consider that these two primordia here are from our bracts primordia, we can see that yes, there is an apical expression in the bract primordia, in both bracts primordia. About aryl, these stages are too early to see the aryl development, because the aryl start the development in spring as a new forming tissue ring at the base of the ovule on the funiculus. And we know, what we know so far is that there are some Maxbox genes involved in the aryl development, and there are at the beginning of the development. Then we know that there are some we know that the aryl undergone to a process of maturation, ripening. That is, this is very interesting. It's quite similar to the maturation and ripening of fleshy fruit. So there is not only a morphological parallelism, but probably there is also a common maturation ripening mechanism.
Thank you. That's very interesting. Well, we are already at 12:01, and they see one other question is basically similar. And Dennis has here a comment that he has 6 months to ask, but he only remembers, how does a micropyle become completely closed? And if we can be brief, and then we'll wrap it up.
Yeah, yeah, that's a good question, because we could, from our morphology, actually we couldn't really understand the mechanism of micropyle closing. So we know that there are some depositions of high complex sugars like callose, but there is still a process of cell. So the cell go in strict contact, and they almost they seal the ovule from the external environment.
Yeah, well, as you know, even though we have these plants growing in our botanical gardens and in front of us, there's so much still to know, and even from the basic development part, and then even more when you include the gene expression and all of that. And this is I think is part of the importance of this, is to realize how little we know, not only how much we know, but how little we know.
Yeah.
This has been fascinating. Thank you very much. I hope you come visit us soon again, and spend another time. We really enjoy having you in the lab. I know I'm speaking not only for me, but for everybody.
Yeah, thank you. It was a pleasure for me. Yeah.
Yeah, sorry, go ahead.
No, I was saying it was a pleasure also for me to be here with you, and yeah.
Okay, well, great. So I want to remind everybody that if you missed part of this event, or you want to watch the seminar again, the recording has been saved and will be published on the New York Botanical Gardens lecture library in the science section in the next few days. Thank you again for being with us today, and we hope you can join us in future science webinars. Until next time, please stay well and safe today, especially with this weather. And thank you again, Sebastiano, for a great seminar.
Yeah, thank
Bon
you.
voyage.
Grazie.